Okay, good afternoon, everyone. My name is Thomas Pons. I'm a physico-chemist researcher at ESPCI, which is an engineering school in Paris. And first of all, I'd like to thank Adrien, Luca, and all the organizers of these study days for the kind invitation and for giving me the opportunity to tell you a bit about fluorescent nanomaterials, and especially about what we will be calling quantum dots. And so you'll see during this presentation how we can obtain these beautiful fluorescence colors using these novel fluorescent nanomaterials. And very briefly, I will detail what is fluorescence through the edges, what are exactly those quantum dots that I will be talking about. And I will detail a few applications, for example, in lighting and displays, and in biological imaging as well. This is a bit far from the exact topic from the study days, but it will open, I hope, some perspectives for some of you. Okay, so first of all, I'd like to emphasize the fundamental difference between lighting and fluorescence with respect to the aspect of the materials. As you know, with lighting, you send some light on an object, some light which has a specific spectrum, and the object then reflects the light minus something that is related to its absorption spectrum. And then if, for example, this object absorbs something that's in the green, you will be left with the blue part of the spectrum and the red part of the spectrum. And so starting from a white light, you get something that has the aspect of something like a purple object. Now imagine that you have an object with a similar absorption spectrum, but that is able to re-emit part of the light that we send onto it as a light from another color. So that's what the principle of luminescence is, or in our case of fluorescence. So for example, here you can see that you can send a light with components on the blue range that will be absorbed by the object and then be re-emitted by the object as orange light. This is a completely different process, and that's why fluorescence objects appear completely different from normal non-fluorescent materials. So fluorescence is basically based on some electronic processes. And here on the right side of the slide are some energy levels represented for the electrons in a fluorescent material by some kind of process that we will discuss in the next slide. This electron will be able to absorb some energy to go from a ground state, initial state, onto an excited state. And then it will be able to go back to the ground state by emitting, giving back the energy as a photon, a fluorescence or a luminescence photon, to go back to its ground state. And here, because after the excitation, you always have some loss of energy due to vibrations or heating of the sample, you always have less energy at the end that you have at the beginning. And so in general, fluorescence materials emit with a wavelength that is redder than the excitation beam. That's what wavelengths it is able to absorb. There are different possibilities to excite the luminescence. You have, for example, electroluminescence, where you send energy, you give energy to the electrons using electricity, and it gives back its energy as in part as light, or it can be photoluminescence, where you excite with light and you obtain light at the end, but with a different wavelengths. And in these two, within this photoluminescence process, you can distinguish fluorescence and phosphorescence, which are slightly different, but not that different. And then you have chemiluminescence. You all are familiar, I'm sure, with these glow sticks, where you mix two chemicals together by breaking a small vial and mixing two reagents react together, and then you have a reaction that provides that ends up with emission of a photon, emission of some light. And also biology also has some kind of luminescence. You have many aquatic or terrestrial animals which are able to emit some kind of luminescence using energy provided by some biological activity, some enzymes, for example, which are able to convert some chemical energy into luminescence used for communication. Okay, so how is fluorescence and phosphorescence? How do these ideas have evolved over time to reach our current era? So, of course, since luminescence is present in many biological or natural phenomena, then for a long time, humans have been familiar with some kind of luminescence, such as those present in aurora or in insects, such as fireflies. But the first real observation of fluorescence, real fluorescence, is reported to be from a guy named Nicolas Monardis, which was who was a Spanish botanist. He actually was studying the trees and the plants in Mexico to give the Spanish ideas about how to take some profit from the local species. And then he observed that a specific kind of wood, a specific tree called the Lignum nephriticum, was able to emit some fluorescence when you were looking at it under daylight. It was giving a blue light, kind of blue tint, which now we know that it's coming from a molecule that is present inside this plant. And you can see here a modern photograph of an extract of this plant, and you can see under daylight that you can see this kind of blue hue that is given, which is due to the fluorescence from the molecules in this solution coming from this plant. So this is the first really historical report of a fluorescent phenomenon. Of course, it was not explained at the time how it worked, and Nicolas Monardis was just reporting that it would be a good idea to look at this fluorescence to avoid counterfeiting, actually, to make sure that you are dealing with the right kind of tree and not a fake one. Then the next report comes a few decades after, in 1602, by an Italian guy in the city of Bologna. This is actually a pretty interesting story. This man was just a worker making shoes, but he was passionate about alchemy. And of course, you know that the main goal of alchemy at the time was to make gold. And so this man was walking in the mountains looking for funny rocks. when he found a rock with a particular aspect, and he brought it with him in his lab, and then he thought that maybe he could heat it to see if he could transform this stone into gold. And of course, it didn't work, because this stone was not gold at all. It was barium sulfate. It was a natural occurring mineral containing barium. But by heating it with coal, unknowingly, by a bit by accident, he obtained a new species that is not a natural species, which is barium sulfide. And of course, he was very disappointed that he didn't obtain gold. But when the story says that when he switched the candle off at the end of the day, he was really surprised, because he could see that the stone was able to emit some light on its own. And this was a phosphorescence phenomenon. And it was really surprising to all scientists for many centuries who were not able to explain this natural phenomenon, that the stone seemed to be able to absorb the light from the sun during daylight and then re-emit it after some time as a reddish light that you were able to see in the dark. And now we know that it's due to some phosphorescence effect. You see that 3 centuries go, and we still have some scientists discovering some phosphorescent minerals, such as this British gentleman, Edward Clark, who was in the county of Durham in Great Britain. And he found some very interesting minerals, some very pretty minerals, which are able to emit some blue and some green light. And this is due to doping of this material with some lanthanides ions, which are able to fluoresce. And these are europium and samarium, which gives this blue and green emission. Of course, the 19th century is really when science, modern science, gets starting. So that's where everything happens. And so George Gabriel Stokes, another British gentleman, was able to study a little bit more in detail the phenomena of fluorescence. And he found out in particular that fluorescence materials were some molecules or some rocks were able to absorb some light and re-emit light at other wavelengths with other colors, but that the wavelengths of the light that was emitted was always longer, redder than the light that was being absorbed. And he studied in particular quinine, which you can find in nowadays in Schweppes bottles. In a few years after that, Becquerel, a French scientist, invented the fluorescence tube. So it's been 150 years, but we're still using fluorescence lamp nowadays. It's a very modern technology, very current. And so it was one of the first real useful application of fluorescence. A few years later, the German chemist started synthesizing artificial fluorescent molecules. One of the first success was the synthesis of a molecule called fluorescein, which gives a green fluorescence, and which is very common. Fluorophore, even still today, can be used as a fluorescence tracer, for example, to study water flow. You put some fluorescein at the entrance of water flow, and you see where the color gets out to try to understand how water flows are connected together. And this guy is named Bayer. He founded one of the very prominent chemist companies, which is still very active today. And then many scientists worked on the synthesis of fluorescent molecules up to 1940s, where they started to bind these fluorescence molecules onto biomolecules to start doing biomolecular detection of specific proteins, for example, to look at where these proteins are located within cells. Here you can see a nice modern example of this, where the skeleton of a cell is labeled with a green dye. And this technology gave rise to a large field that is very common in biology, that has enabled greatly the expansion of biology, of cellular biology, more specifically. Up to just a few decades ago, with the use of another fluorescent materials, which is not a molecule, but a biomolecule, a naturally occurring protein, um, which comes from a fluorescent algae, and that you are able now to introduce in some genetically modified animals or cells, to be able to study in very fine details biological phenomena. This discovery has earned three scientists the Nobel Prize a few years ago. Fluorescence is very present in our everyday life. It goes from highlighters, of course, very easy examples to give of fluorescence objects, but it's also present in lamps, in lighting, for uh, energy efficient lighting. Fluorescence is very well used because you can very efficiently create first some electroluminescence from a gas inside the lamp. And this electroluminescence, which is usually in the UV, is then absorbed by fluorescent materials, which is the one that you can see within this white coating inside the lamp, and which is then re-emitted as visible light. Very efficient. It's also present in washing powders, for example, because if you want something that is very white and brightly white, you need all the colors from the spectrum. And unfortunately, when the tissues are aging, very often it starts to take this yellowish color, and so to gives back its very whitish aspect, you need to add some blue light into it. And so you have blue emitting fluorophores in these washing powders that gives the consumer the impression that the tissue is becomes white again. And another final example, very simple example, is, for example, the detection of fingerprints in forensic science. So you can see that fluorescence is everywhere within our houses and so on. Okay, so it goes back from the 16th century with a lot of progress that we can see up until now. And the fluorescent materials that I will talk about in these presentations are fluorescence materials, fluorescence nanomaterials. Here you can see that we started from natural phenomena, and then man discovered luminescence within these inorganic materials, within these stones. And then we moved to organic materials, which means fluorescence from small organic molecules or biomolecules. And now we are getting back to inorganic materials, so like stone, but not as a centimeter or millimeter scale, but in the nanometer scale. So our quantum dots are fluorescent nanocrystals. By this, I mean that they are tiny crystals. Here, this is an electron microscopy image of such a crystal, in which you can see that all the atoms are arranged in a very precise way, a periodic way. This is why it's called a crystal, and it's a nanocrystal because it's only a few nanometer across, a nanometer being a billion times smaller than a meter, which means that it's typically a few 10 or 20 or a few tens of nanometer of atoms across in diameter. So it contains typically a few hundreds to a few thousands atoms. So we are at the scale that is just above the scale of the atom. We are putting a few hundreds typically of atoms together, arranged in very particular way. And we are choosing these atoms to form materials that we are calling semiconductor. And I will explain just in a minute what I mean by that. How do we make these nanocrystals? Typically, there are two main strategies to fabricate nanocrystals. You can start from the massive bulk material, which is, for example, at the centimeter scale or more. And you can break this very large material into very small, tiny particles. This is not very efficient because you never control precisely the size or the shape of the particles that you break, that you create. Instead, what we are doing is what's called the bottom-up approach, meaning that we start from the components of the materials, from the atomic precursors. For example, if these nanocrystals contain two different species of atoms, then we will introduce precursors for each of the species present in the final crystal, and we put them in a solvent, and then we heat everything at typically temperatures between 200 and 300 degrees Celsius. And then after a few minutes, these precursors start to react together, and under very precise control conditions, we're able to form these tiny nanoparticles, these tiny crystals. And we will see in a moment that we can choose the materials and the size of this nanoparticle in order to have particles that emit fluorescence at different wavelengths to create this nice rainbow of fluorescent solutions. So at the end of the process, we obtain nanoparticles which are suspended in a solution. We cannot see them because they are too small, but they are completely homogeneously dispersed in these solutions, which can be organic solvents or water or polymer matrices or some other inorganic materials and so on. Okay, so what happens in our semiconductor? Typically, if I take the example of cadmium selenide, it's composed of cadmium atoms and selenium atoms arranged together in a crystal, extending in all directions. Here, the selenium atoms are represented in yellow, and you can see that it's represented at selenium 2-, which means that they have 2 extra electrons around these atoms. Cadmium is in gray. It's represented at cadmium 2+, which means that it lacks 2 electrons. And so these extra electrons from the selenium initially were on the cadmium. But in the ground state, in the initial state, the state of lower energy, the electrons are located on the selenium. And this material is a semiconductor, which means that if you give it enough energy, you will be able to transfer 1 electron that is on the selenium atoms onto a cadmium atom. For this, it needs to absorb some energy because this new state is not energetically favorable. It costs some energy. And then it will be able to give back this energy as a luminescence, so as light. And typically for cadmium selenide, this would be a very red light, so really at the edge of the visible spectrum, around 700 nanometer or so. Okay, so that's a very simplified version, because actually in the almost infinite crystal of cadmium selenide, the electrons are not really located on a specific atom of selenium or a specific atom of cadmium. They are distributed among a few tens, typically a few hundreds, of atoms, of neighboring atoms within a specific region, spatial region of the crystal, which means that the electron here is distributed among all these atoms. So it likes to extend, um, to avoid being located at a specific point. And what we are doing when we are making nanocrystals is that we are restricting the crystal to a size which is below this natural size into which the electron likes to extend. And so we are forcing the electron to remain located in a small crystal that is smaller than its natural size. And this is the domain of quantum physics. So quantum physics is actually a bit scary, but I will show you that it's very easy, and you know everything that you need to know about quantum physics already, because every one of you know the guitar, right? So the guitar is, you may know or you may not know it, but it's a quantum instrument. Because when you strike a chord, you see that you create some vibration on this very short time acquisition picture. You can see that the vibrations on each specific chord, they are not completely random at all. They have a specific wavelength. Why do they have a specific wavelength? That's because the chord has a fixed length. And so the chord cannot move at either end at its ends. So here it's fixed. And so the vibrations have to be at a certain frequency so that the displacement of the chord is zero at both ends. And so a fixed length means a fixed frequency. And then when you reduce the length of the chord, you reduce the wavelength. And this means that you increase the frequency, which means that you have a higher pitch, for example, in the case of the guitar. All right, so everybody is familiar with this. And in the case of quantum dots, it's exactly the same idea. You are restricting the electrons to vibrate in a fixed volume. And just as in the case of the guitar, a fixed length, a fixed diameter, if you like, of a nanocrystals means a fixed frequency. And so the electrons needs to vibrate at a specific frequency depending on the length of the diameter of this nanocrystal. And so it will emit light with wavelengths that corresponds to, that is fixed by the size of the crystal, right? Because wavelengths are a specific wavelengths correspond to specific energy, and you have determined this energy with the frequency of vibrations of the electrons. Now, if you reduce the size of the nanocrystal, you reduce the wavelengths, you increase the frequency, and so you shift the light that is emitted to a different color, to a shorter wavelength. So, for example, if you take the same material, cadmium selenide, and you make nanocrystals that are 2 nanometer in diameter, for example, or 2.5 or 3, 3.5, and so on, and up to 5, each size will emit a different wavelength, a different color. And what is very interesting with the methods that we are using to fabricate this nanocrystal is that we are able to obtain in one set of conditions nanocrystal inside the solution that all have the same size, and so they will all emit with the same color. Now, when we change the material, we can change the range of wavelengths that we can reach while making nanocrystals. And so this is a visible range here from something like 400 nanometer to 700 nanometer in wavelength that we can pretty much cover with a material like cadmium selenide, but we have other materials that work as well, such as indium phosphide, which is made of less toxic materials. And we can also have materials that emit in the ultraviolet or in the near infrared in below or above the visible range. Okay, so why are these materials interesting to us? That's because compared to organic fluorophores, to organic molecules, these materials are very bright and they emit light very efficiently, which means that from an energetic point of view, you don't lose a lot of energy when you give it some energy and then it gives back this energy as fluorescence. The second point is that it's an inorganic material, which means that it's very stable. Think of it as a small piece of rock. It's very hard, very stable. Nothing really can happen to it. So it's able to emit light for a long period of time. Then, because we are able, as I was saying, because we are able to fabricate nanocrystals that all have the same size, they will all emit with the same color. So they will have, in other words, a narrow emission spectrum. We are able to tune this emission from the ultraviolet to the near infrared, and it's pretty cheap to fabricate in comparison to other semiconductor materials. And so I will show you that we can design some specific applications, for example, in displays or for biological imaging. First example I will show you is the use of quantum dots for displays. And here we will mainly use the fact that we're able to obtain solutions that emit light with very pure colors, which means that if you take an emission spectrum, the width of the spectrum will be much narrower than what you would get with other kind of fluorescent materials, such as inorganic phosphors or organic molecules. This is used nowadays in televisions because televisions, LCD televisions work because they have a blue light that emits light in the blue range with a very efficient energy conversion. And then this blue light is partially absorbed by a phosphor, which converts this blue light into green and red light. And then you have filters to obtain the three channels, blue, green, and red. But the problem with the standard inorganic phosphors used in television is that this spectrum is very broad. And so if you look at the color obtained through the green spectrum, it's not through the green filter, it's not very pure green. It's actually a mixture of green, bluish green, and yellow. If you look at what you obtain in the red, it's also a mixture of orange and red. So if you represent these colors on the color diagram, you will get these 3 points for the red, green, and blue. And by mixing these colors, you are able to obtain all colors within this triangle. Now, with quantum dots, you can define emitters that have a much more narrow emission spectrum. And so the final colors that you obtain through the green filter and through the red filter are much more pure than what you had with the standard TV. And so now you shifted, you shifted this green point towards something that is greener, and this red point here towards something that is redder. And so you obtain a much more extended range of available colors. Here, if you compare an example, exaggerated example, of course, for a commercial product, this is the same image that is supposed to be shown on a standard TV screen and on a TV screen containing quantum dots. Here you can see that the green of the frog and the leaf in the background appears a bit yellowish, and here you're able to see something much greener. So in terms of color, we have something that is purer, and so gives a display with a higher fidelity. Another example of application is biological imaging. Okay, so we have all these emitters that are very bright and very stable. And we can have some emitters that emit in the visible part of the spectrum, some other emitters that emit in the near-infrared part of the spectrum. We will use these nanocrystals emitting in the visible part to image biomolecules within cells. And because they are very stable, we will be able to observe even a single emitter. And we will use that to track down the motion of single molecules within cells. A typical example of applications that I will just explain to give you a more specific idea of what we can do is the tracking of neuron receptor. So you imagine that you have a neuron here. This is a membrane of the neurons. Here is the outside, here is the inside. And the neuron communicates with other neurons and with its environment within the brain. And that's how the brain works, using these small proteins that are within the membrane. And it will form a receptor. So for example, here it's an example where we wanted to study a cannabinoid receptor. So when cannabinoids are in the environment, it binds to this receptor, and then it activates it, and then it does something inside the neuron. It creates some biochemical reaction, or it can also create the motion of this receptor. If this receptor is outside a synapse being this location where two neurons interact together, it's not really a big deal if this receptor is affected. But the neuron is able to move these receptors around as a function of its activity, to move it towards the synapse, to make it active and have some effect on memory, for example, or on just the way you think, or to the cell nucleus, or to inside the cell, as opposed to at the membrane, to recycle this receptor and either destroy it because it's not needed anymore, or to recycle it and put it at some other place. And so a very interesting question is trying to understand how this receptor move around at the membrane of the neuron as a function of the activity and as a function of the exposure to, for example, cannabinoid. To do that, what we do is we fabricate one of these quantum dots, and we bind it to an antibody, which is a specific molecule that is able to recognize very specifically this membrane receptor. And then we will use a fluorescence from this small quantum dot to image the cell and to see where is this receptor. In practice, it looks like this. You can see that this is the body of a neuron. It has many dendrites and axons that extend out towards other neurons. And this green fluorescence is from another molecule that's just here to see where the neuron is. And these small purple dots, each of these purple dots are representing the fluorescence emission from one quantum dot that is bound to one receptor. You can see that with time, some receptor will remain at the same location, some other receptors will move along the axons, and now we are currently studying how this behavior evolves with exposure with cannabinoid to try to understand the phenomena of addiction, especially in young brain as opposed to adult brains. Okay, so this was an example using visible emitting quantum dots. Now, infrared emitting quantum dots are also very interesting because if you want to look at whole body, for example, to try to understand cancer biology or developmental biology, or to try to image pathologies within a patient, know that visible light doesn't go through the tissues very well, doesn't go through the body very well. But infrared is much more efficient. If you were able to see with your eyes in the infrared, you would see actually not the surface of your skin, but you would see up to a few millimeters or a few centimeters deep within your body. That would be a bit strange. But anyway, we are able to use these infrared emitting quantum dots to image, to do some what we call in vivo imaging. For example, if we inject these infrared emitting quantum dots in the bloodstream of a mouse, this is how it looks like. You don't see the surface of the mouse anymore, but you see everything that's inside without having to harm the animal. You don't have to cut it open. You just have to put it under an infrared camera, which is able to detect this light in the near infrared. And you can see that you see the blood veins and arteries deep within the animal. And you can see the heart beating and the liver and so on, and many fine details or where these quantum dots are circulating within the bloodstream. Now, it makes nice images, but it's even more interesting to use that for a specific purpose. And now we are trying to use that to understand better cancer biology, for example. This is a mouse that's bearing a tumor in its brain. And people can use these images here. These images are from blood vessels within the brain of the mouse. Again, you don't have to cut open the brain of the mouse to image, to take these images, which is very nice. You just have to locate a CCD camera and magnify the image, and just image across the skull of the animal without having to open it. Here you see the normal vasculature, and things move around very fast, very quickly. And you can observe that around the tumor, the vasculature is much more disordered, and the blood flow is much slower, which is something very important to understand in the context of cancer biology and being able to design new treatments to treat cancers. Okay, so we'll finish with this, and I hope that you understand a little bit better what these fluorescent nanomaterials are. They have been blessed with 3 gifts: gifts of brightness, stability, and versatility. You have seen that they are being used for many applications. And so they have certainly a bright future as fluorescent materials. We just have to better understand how to make them stable over a very long period of times, not just minutes of hours, but probably days or years if we want to use them as efficient lighting sources. And also we have to learn how to recycle them better to avoid pollution and toxicity issues. And I will finish with this, and I thank you for your kind attention, and I will be happy to answer any question if you have any. Thank you.